So, how do genes work? How does a series of molecules, the A's, the C's, the T's, and G's, in certain order, lead to something as profound as different eye colors or, balding early in life or any of a number of things? How do we get from the genotype, the genome, all of the genes contained in an organism, to the phenotype, the physical manifestation of the instructions? And the way that this happens is through a process called transcription and translation. Transcription is the sequence for a gene is copied. And it's copied from where? From the DNA molecule that sits inside the nucleus to a middleman molecule called messenger RNA. Then that messenger RNA molecule leaves the nucleus and enters the cytoplasm, where it undergoes translation. And translation is where we actually take the genetic code found in the DNA and mRNA and translate it into a functional protein sequence. The analogy used in my lecture here and in a previous textbook that we use is like a cookbook. And I think it's actually a decent analogy that you might discover your grandmother's old cookbook or your mother's old cookbook, and it has a recipe for cookies in it. And, you just have a little note card and there's all sorts of extra notes and stuff in the cookbook. So you don't write down the instructions exactly. You transcribe them, you rewrite them, but you rewrite them in a slightly different version that will allow you to still follow the instructions, but using your own sort of words and sentences. You then take that index card with the recipe and read that and translate that into the actual cookies by going and getting the ingredients and putting them together. And the end product, the cookies, is kind of like the protein molecule. So I think it's an okay analogy that sort of describes this process. You're taking words on paper and ultimately turning them into cookies that end up in your belly. So take home message. The genes and strands of DNA are a storehouse of information. They're an instruction book. They're a cookbook. The process by which this information is used to build an organism, to go from genotype to phenotype, occurs in two main steps that we'll talk about more in 1 minute. Transcription is where a copy of the gene's base sequence is read, made, and transferred so that we can then proceed to translation, in which that copy is used to direct the production of a specific protein. Okay, so building organisms, information in DNA directs the production of the molecules that make up an organism. So we're going to start with transcription. And this is simply reading the information coded in DNA and sort of rewriting it in a slightly different way. There are 4 primary steps to transcription, labeled 1, 2, 3, 4 here on the slide. But even before that, we must first unwind and rewind a section of DNA. Well, first we have to unwind it. We'll rewind it after we actually do the transcription. So there are enzymes that can land on here and unwind the molecule. And likewise, there are enzymes that will facilitate the rewinding of the molecule. And that class of molecules, of enzymes, are called helicases. Now, the enzyme that will actually be producing our mRNA or messenger RNA molecule is an enzyme called RNA polymerase. A polymerase is any molecule that's going to make a nucleic acid. It could be DNA, RNA, whatever. So RNA polymerase obviously makes RNA. Now, the first thing that the RNA polymerase molecule needs to do is land on the correct spot of the DNA molecule where a specific gene is going to start. And it turns out that genes have evolved to have a specific place right before the gene starts that is recognized by the RNA polymerase, and it's called the promoter region. And the promoter region is just a sequence of base pairs, just like anything else, that the RNA polymerase can preferentially bind to, likely because of hydrogen bonding and the folding of the protein. but it lands right before the start of the gene. And we say that it is landing upstream. You can think of DNA like a stream. So upstream of the gene would be before the gene starts. Downstream of the gene would be after the gene. So step one is that the RNA polymerase molecule has to recognize the promoter site and then bind to one strand of the DNA molecule. And then it begins to read the gene's message. Now I'm going to include a video on Canvas under the lab section that actually shows an animated version of this process because it's pretty awesome. And once recognition and binding has occurred, then we can go to step two, which is transcribing or transcription. And this actually happens extraordinarily fast. So the molecule moves down the DNA template very, very quickly. As the DNA strand is processed through the RNA polymerase builds a single-stranded copy of the gene called the messenger RNA transcript. And remember, this isn't that tough because DNA has complementary base pairing. The only difference here, if you remember, is that there is no T in RNA. So instead of having a T where there's an A in DNA, you have a U for uracil instead. So there's no T's in the mRNA transcript, only the U. So after recognition and binding, we have transcription. And you can see what's happening here is as the RNA polymerase is moving down the DNA molecule, it's taking free base pairs that are in the nucleus and adding them to this mRNA transcript right here. And what you see coming out the end is this growing mRNA molecule. Now, just like there's a recognition site, a promoter site at the very beginning of the DNA gene, of the gene, there's an end portion as well. And when the RNA polymerase reaches a certain set of base pairs at the end of the gene, it falls off, and that's called termination. So when the RNA polymerase encounters a code, a series of base pairs signaling the end of the gene, it stops transcription and releases the mRNA transcript. The last step is capping and editing. Oftentimes, before the mRNA transcript can be translated into a protein, a cap and tail are added for protection because it's going to leave the nucleus and enter the cytoplasm. And those non-coding regions, the introns that are within the gene, are often removed. So what you have is just a short transcript of the specific coding section of a gene, but instead of DNA, it's now single-stranded RNA. That's it. Now it will leave the nucleus and enter the cytoplasm. So remember the steps. We're going to unwind. We're going to recognize and bind where the RNA polymerase molecule lands on the promoter region. We will then transcribe the DNA into an mRNA transcript. Once we reach a certain code at the end of the gene, that will signal termination and the RNA polymerase molecule falls off. Finally, there is capping and editing of the mRNA transcript, and then it leaves the nucleus and enters the cytoplasm. Now, just to refresh your memory, we can ask which answer below represents the mRNA strand copied from the DNA strand below. So this is DNA, so we should expect to see T's. But we can immediately get rid of two of these answers because we know we're making an mRNA transcript and there is no thymine in mRNA. So it can't be 3 and it can't be 4. This makes it easy. Everywhere there's a T, there should be an A in the mRNA transcript, so we can get rid of that guy. And that means that we're going to have number 2 be the answer. I'll let you double check to make sure that this is transcribed correctly. Okay, take-home message. Transcription is the first step in the two-step process by which DNA directs the synthesis of proteins. In transcription, a single copy of one specific gene within the DNA is made in the form of a molecule of messenger RNA, or mRNA, which moves where it can be translated into a protein. Okay, next we will focus on translation. Translation is where that mRNA transcript, that copy of information from the DNA, is actually used to build functional molecules. And this takes place inside of a ribosome. We've talked about ribosomes before, but they are the organelles that are the site of protein synthesis. And you can see that they themselves are just very complex proteins. The 30S subunit implies that there are, in fact, two different proteins that come together to make the functional ribosome. And that's true. They kind of make a little sandwich around the mRNA transcript in order to translate the mRNA transcript into a functional protein. So we need several ingredients in the cytoplasm for translation to occur. We need free amino acids, that is, amino acids that are not already part of a protein chain. We need ribosomal units because this is going on all the time. So oftentimes ribosomal subunits are already occupied making proteins. And you need something called transfer RNA. So you need another RNA molecule that is really critical in translating the mRNA sequence. This is what transfer RNA looks like. You see over here, it's held together with hydrogen bonds, and we still have some of that complementary base pairing. On one end is an amino acid, and depending on which transfer or tRNA molecule we're looking at, that amino acid will vary. Transfer RNA molecules translate the mRNA code. They link specific groups of bases of ACs, U's, and G's on the mRNA with specific amino acids that will be used to build a protein. And it turns out that the number of bases that correspond to a specific amino acid is 3. So each set of 3 bases on the mRNA transcript or on the DNA template itself is called a codon. And each codon corresponds to a specific amino acid. So you have these tRNA molecules floating around with a specific amino acid, and on the other end, they have a region called an anticodon, which binds specifically, because again of complementary base pairing, to a specific codon on the mRNA molecule. So how do you know which amino acid is supposed to go where? Because every tRNA molecule, again, has a specific region on one end that corresponds to a specific 3-base region on the mRNA called a codon that is unique for a specific amino acid. So, how do we know which codons code for which amino acids? Well, we actually have a table that will tell us. This is called the genetic code, and this is sort of like our Rosetta Stone, right? That allows us to figure out what the bases in DNA actually produce in terms of a functional series of amino acids. And the way that you can read the genetic code, the way that you can read a sequence of DNA or an mRNA transcript is by simply doing the following. This first column implies the first position in one of those 3 base pair codons. So let's say that I tell you there is a 3 base pair codon of CAG. CAG. Okay. We come here to where it says first position, and we said the first position is a C. So here's U, here's C. So I'll stop here because we don't need to go to A or G. So we know we're going to be looking in this row right here. Okay. But we still need some more information. We need what is at the second position. The second position is labeled across these columns right here. So we said CAG. So here's our C's. Here's U at the second position, C at the second position, A at the second position. So we know that we are CA something. So we've narrowed it down to the amino acid either histidine or glutamine, all right? But we said that we are CAG, so now we need to come to this third position, which is over here. And remember, we're down here, so we got to go UCAG. So first position, second position, third position. Here's CAG. Oops, sorry. Here's CAG right there. So the codon CAG codes for glutamine. And there is a tRNA molecule floating around with an anticodon for glutamine. And everywhere that tRNA molecule finds CAG on a piece of mRNA, it's going to deposit a glutamine amino acid. Let's say you then encounter another codon that says ACC. We can figure out what that makes. We'll come here to the first position, A, then the second position, C, so we know we're right here. And notice that it doesn't matter whether we have a U, C, A, or G in that third position, we're going to make threonine. So it turns out that there is some redundancy here, that oftentimes, no matter what the third base pair is, it produces the same amino acid. Now, you'll notice that there are a couple of interesting codes here. Start and stop. Start and stop. Start and stop are specific codons that tell the RNA polymerase this is where the gene starts and this is where the gene stops. So the very first amino acid in almost any protein is going to be AUG because that tells RNA polymerase this is where this gene starts. This is where I know where to begin transcription. Likewise, there's a region here, let me take a step back. This AUG here, methionine, doesn't tell RNA polymerase where to start. It tells the ribosomes where to start. And likewise, these codons here, UAA, UAG, UGA, these are called stop codons. This tells the ribosomes, all right, you're done translating and the protein is finished. So those are very important as well. So translation only really has 3 steps. And again, this is a much more complicated process than we're making it out to be, but I just want you to know the basics of this. The 3 steps are recognition and initiation, elongation, and termination. Let's start with recognition and initiation. Recognition starts with a start codon, that AUG. This is important because it tells the ribosome, all right, we are going to take our 2 subunits called the large and small subunit and sandwich it right over this start codon, and that tells us which amino acid will start this protein. And it's always going to be methionine, always. So that is called the recognition site. That tells the ribosomes where we're going to start. Then we have elongation. Several tRNA molecules can fit inside of a ribosome at the same time. So the ribosome is moving down this mRNA transcript right here. You have the free tRNA molecules floating around. And what happens is as a tRNA molecule finds the correct codon on the mRNA sequence, it binds and then moves into the ribosome. Once in the ribosome, it adds its amino acid to the growing protein chain before moving along and then getting kicked out to go bind to another amino acid. And once this one is kicked out, the next one can move in. So that's all elongation is. As the ribosome moves along the strand of mRNA, each new amino acid carrying tRNA molecule binds to the next 3 bases on the mRNA transcript. After it is attached the growing protein chain to the new amino acid, the tRNA is kicked out and detaches and floats away. Finally, at the end of every mRNA transcript is one of those 3 stop sequences. And that stop sequence tells the 2 subunits of the ribosome, we're done, we can detach from the mRNA transcript, and we can release our newly formed protein. Now, this does a couple things. One, that protein, depending on where it's made, will move through one of the endomembrane organelles, like the rough endoplasmic reticulum or the Golgi apparatus, and be packaged for movement somewhere else in the body. The ribosomal subunits are then free to go find another mRNA transcript, and the mRNA transcript itself can be reused. There's nothing wrong with it, and in fact, it can be used dozens or even hundreds of times before it starts to degrade and new ones have to be made. The take-home message. Translation is the second step in the two-step process by which DNA directs the synthesis of proteins. In translation, the information from a gene that has been carried by the nucleotide sequence of an mRNA molecule is read, and ingredients present in the cell's cytoplasm, ribosomes, free amino acids, tRNA molecules, are then used to produce a protein. Okay, I think we'll stop that video right here, and then we'll talk next about how cells regulate transcription and translation.